Hello, today we're going to cover Chapter 4, the action potential. And just a quick review from our last class session, remember that ions move across the neuronal membrane through different gradients. We also have different channels that are selectively permeable to particular types of ions, such as sodium ion channels and potassium ion channels, and they can diffuse across the concentration gradient via passive transport. Most ion channels are only selectively permeable to sodium or potassium and will only allow specific ions to enter or exit the neuron. But remember, there's another form of transport we've talked about in class known as active transport. This is when ions are going to move against their concentration gradient. And because they have to move against their concentration gradient, they must utilize energy provided by adenosine triphosphate, or ATP. So here we have sodium coming inside of the channels, and this is going to depolarize the cell. This actually makes it more positive inside of the neuron, when at resting potential we know the inside of the neuron is going to be more negative. So we know that ion channels can stay open for passive transport, although some require signals to open. Remember, we talked about two different types of channels, including ligand-gated ion channels and voltage-gated ion channels. Now, we know that ligand-gated ion channels require a certain neurotransmitter of interest to combine to the receptor, which will allow conformational change, essentially meaning that the ion channel gates are going to open and allow ions to come in and out of the cell. We also have voltage-gated ion channels, as we've talked about before, in which these channels are selectively permeable to particular ions based on membrane potential. When the membrane potential reaches the value that's necessary for this channel to open, it allows ions to move in and out of it. Here's a fun fact about sodium channels. There are certain drugs that can actually affect the ion channel function by binding to the channel protein. It can actually alter its function. This is the case with lidocaine, which is an anesthetic, and this is a good example of how a drug can actually directly bind to voltage-gated sodium channels and block the channel, thus inhibiting sodium entry into the cell. As ions move through their particular channels and crossing the cell membrane, they cause the membrane potential to move away from its resting potential. Now remember, we've talked about how a cell's resting potential is typically negative 65 to negative 70 millivolts. However, when the cell is not at rest and it's inducing a nerve impulse known as an action potential, the cell is going to become depolarized, meaning that it's going to become more positive inside of the neuron relative to the outside of the cell. If this is just a small change, it's called a graded potential. They typically don't result from the opening of voltage-gated channels. Graded potentials are very small and allow the neurons to reset. And a great example of this is the sodium-potassium pump. The sodium-potassium pump can actually regulate and accomplish these small graded potentials. Remember, this is when there is active transport of 3 sodium ions outside of the cell and 2 potassium ions back into the cell. This pump is restoring the electrochemical gradient and allowing a neuron to rest. Remember that this pump, the sodium-potassium pump, is working constantly and simultaneously with other ion channels on the cell membrane. And it's going to help to ensure that the cell can rest by allowing that influx of the 2 potassium ions back inside of the channel into the cytosol. As you should know by now, at rest, there are more positively charged ions outside of the cell relative to the inside. And this difference in charge is known as the electrical gradient. If we look at the difference in total charge inside of the cytosol of the neuron versus the charge in the extracellular fluid outside of the cell, this is known as its membrane potential. And here's a great example of membrane potential. As you can see here, there are more potassium ions present inside of the cytosol, and there are also organic anions that are contributing to its negative charge. Whereas in the extracellular fluid outside of the cell, we know the main ingredient here is water, but there are these positively charged atoms known as ions. Remember, there can be positive ions known as cations and negative ions known as anions that are going to be contributing to this charge outside of the cell and inducing it to be more positive at rest. Remember that at resting membrane potential, a cell's voltage is typically around -65 or -70 millivolts. This is going to be important as we learn more about action potentials. The concentration gradient and the electrochemical gradient for every ion is equal and opposite inside and outside of the cell, thus contributing to resting membrane potential. So this essentially means that the inside of the cell is 70 millivolts less positive than the outside of the neuron. And remember, at rest, the neuron has more potassium inside relative to the outside. When a cell is not at rest, an action potential, also known as a nerve impulse, can occur. So what exactly does this mean? Essentially, the action potential is a rapid reversal of the resting membrane potential. Remember that the typical neuron has an average resting membrane potential of about -65 to -70 millivolts. Thus, the action potential is a rapid reversal of this, meaning the cell becomes more depolarized or more positive inside of the neuron relative to the outside. So the inside of the membrane becomes depolarized, which causes this nerve impulse. Here at the axon hillock, this is the very beginning of the axon proper. If you remember, this is the part that is closest to the soma or cell body of the neuron. Now, we've talked a lot about ion channels and the sodium-potassium pump and how they're embedded in the phospholipid membrane of the cell. These channels are highly concentrated in the axon hillock, and thus, if this area becomes depolarized, it will initiate an action potential or nerve impulse that is submitted and pushed down the axon across the entire axon proper to the end of the axon known as the axon terminal or the terminal bouton. Action potentials are different from graded potentials in that they do not diminish in size or frequency. Once an action potential begins at the axon hillock, it will jump from node to node across the myelin. These are known as the nodes of Ranvier. And this is super crucial because synaptic transmission or cell communication couldn't occur if action potentials diminished and did not reach the axon terminal. This is really important when we're understanding electrical synaptic transmission. And remember that this is different from chemical synaptic transmission, in which cells are transmitting neurotransmitters at the synaptic cleft to one another. We're going to talk more about that in our next couple of chapters. Right now, I just want you to focus on electrical neurotransmission and how ion channels can induce this depolarization in the cell, which causes action potentials to jump down the axon proper. So where do action potentials travel? We know that the axon of a neuron is myelinated, and this means that it is essentially insulated. The myelin sheath is this protective coating that increases the thickness and increases the diameter of the axon. This is important because myelin does help conduct faster nerve impulses. Remember, there are two different types of cells that create myelin, and you'll need to know those for your exam. The first is oligodendrocytes, which are a glial cell in the central nervous system that helps to create myelin. They typically sit around the neurons. And in the peripheral nervous system, there are Schwann cells, which work in the same way that oligodendrocytes do, except they're in the peripheral system instead. So those are the two types of glial cells that are largely creating myelin. Additionally, we know that the axon is bathed in a salty extracellular fluid, and these positively charged atoms or ions are what induce this electrical charge in the cell. So both of these are contributing to the action potential. So keep in mind this big picture. The action potential is essentially like a frequency or a pattern that constitutes a code, and neurons can transfer this code from one another. Essentially, this is a transfer of information. Action potentials begin at the axon hillock, but they travel down the entire axon. And just a reminder, voltage-gated ion channels in the axon hillock are highly concentrated, and thus this is where an action potential typically begins. Here's an example of an action potential in a pyramidal cell. So remember that pyramidal neurons can be typically found in different brain areas, such as the hippocampus or the cortex. This pyramidal cell has plentiful dendritic spines, and the spike initiation zone would typically be in the axon hillock right below its soma or cell body. This membrane here along the axon proper has a high density of voltage-gated sodium channels, which will induce depolarization or increased positive charge inside of the neuron. So I wanted to show you guys an action potential in a real neuron. This was a really cool experiment in which neuroscientists actually used a fluorescent tag inside of real neurons. So these neurons were taken, they're primary neurons that were taken and plated in a petri dish, and they were cultured. And so as they were growing these neurons, they added this fluorescent tag so they could look at the action potential in real time. So let's take a look. As you can see, this action potential occurs so quickly, it happens in a matter of milliseconds. So now we're going to go over each step of the action potential, and hint, hint, you might have to label this for your exam. So make sure you really understand this. First, there's the rising phase, which is also known as depolarization of the cell. And this is when sodium ions largely come inside of the neuron to make it more positive. The second step is the overshoot, when the cell reaches its maximum internal positive charge. In this case, there is a maximum amount of sodium ions that have entered the cell, and it can no longer increase its positive charge. In the falling phase, known as repolarization, the sodium ion channel gates close and the potassium ion channel gates open. In this falling phase, it's important that there's an efflux of potassium ions moving outside of the cell to help the cell regain its negative charge. Remember, the negative resting membrane potential inside of the cell is largely induced by the efflux of potassium ions, or in addition, usually simultaneously, the organic anions that remain inside of the cell's cytosol. So both the efflux of potassium and the organic anions inside of the cell are going to help it repolarize or become more negative. There's also the undershoot that's known as hyperpolarization, and the cell becomes even more negatively charged than it did at rest. Now, this is because potassium may still be leaking out, contributing to this negative resting membrane potential. If you remember, neurons typically have more ion channels that are selectively permeable to potassium rather than sodium. And this is important because potassium needs to be able to leak out of the cell to help the cell remain in its resting state. And that is why there are more ion channels that are more selectively permeable to potassium rather than sodium. So now let's go through this step by step with these images. So first we're going to look at the threshold. The threshold of the action potential is when the cell becomes slightly more positive. So first, This is the resting potential at about negative 65 or negative 70 millivolts. Now we know if some external sodium comes into the neuron, this is going to go through a selectively permeable sodium ion channel, and this small influx inside of the cytosol of the cell is going to make it more positive. If the cell reaches about negative 55 millivolts in its cytosol, this is when an action potential can potentially occur. During depolarization, the firing frequency of action potentials reflects the actual magnitude of the depolarizing current. So as you can see in this image, that stimulation intensity is encoded in the nervous system. And here we have the overshoot. So once depolarization occurs and sodium ions influx inside of the neuron, there's this moment called the overshoot in which it reaches its maximal positive charge at about 30 millivolts. At this time, the sodium ion channel gates will close and the potassium ion gates will open. So remember that sodium can no longer enter the cell at this overshoot. So after these sodium ion channel gates close, there's something that's known as the absolute refractory period. During this time, another action potential cannot occur in the neuron because all of the sodium ions have already entered the cell and it's currently too positive for another action potential to occur. This is usually at about positive 30 millivolts. If you remember, action potentials can't usually occur until they reach a threshold of about 55 millivolts. This is known as the absolute refractory period because an action potential cannot happen at this time. It absolutely is not possible. And it's after this absolute refractory period at the overshoot in which the potassium gates will open. Let's move on to that next step in the next slide. So during the repolarization phase, we know that the sodium ion channel gates are closed. During this time, the potassium ion channel gates open, and there is a slow efflux of potassium outside of the cell. And because potassium is a positively charged cation, when it leaves the cell, it becomes repolarized, meaning it regains its more negative resting membrane potential. So essentially, this efflux of potassium is going to help the cell get back to its resting membrane potential because it is becoming more negative inside of the neuron relative to the outside of the neuron. Here's what repolarization looks like in real time. So as you can see, we have this green ion channel. This is selectively permeable to the ion potassium. And we can see here that as this ion channel opens up, potassium is going to slowly exit the cell into the extracellular fluid. This is occurring right here at repolarization. When potassium exits the cell, it's going to become more negative because it's a positively charged cation. And this occurs in a matter of milliseconds. Interestingly, after repolarization, there's something called hyperpolarization. The potassium ion channels close slowly. Recall that we've talked about leaky potassium channels and how there are more popular and abundant amounts of these potassium ion channels. So since it leaks out slowly, the membrane can become even more hyperpolarized, meaning that it's going to be even more negative than the typical resting membrane potential at about negative 65 or negative 70 millivolts. This is because potassium is still leaving the cell. During this time, during hyperpolarization, the cell's membrane potential is going to be less negative than its resting membrane potential. As the neuron's charge becomes more negative again, it's possible for sodium channels to open back up, but it would take a very large stimulus to induce another nerve impulse or action potential. During this time, This is known as the relative refractory period. This is when the cell has regained its resting membrane potential, but it has not reached threshold yet. At this time, a very large stimulus would need to activate the cell to allow extracellular sodium ions to re-enter the neuron. Remember that action potentials are occurring all the time in our body. This is a cycle that's occurring to help us move. We have action potentials occur at the neuromuscular junction. If you remember, we've talked about how motor neurons can send nerve impulses down the spine via the ventral root and onto the muscles of interest. We can go back to our example of the simple reflex here. For example, if you needed to move away from a sharp object, you would receive that sensory pain up the dorsal root ganglia of the spinal cord, and your motor neurons would send and transmit nerve impulses back down to your foot via the ventral root. And motor neurons would synapse at the neuromuscular junction to actually cause that reflex and to cause your foot to move. These are nerve impulses or action potentials that are occurring. And essentially, during this relative refractory period, if you were to step on a very sharp object that would function as that really large, strong stimulus, during this time, you could generate another action potential. This is known as the relative refractory period. And for your first exam, make sure you understand the difference between the absolute refractory period up here at the overshoot and the relative refractory period down here during the repolarization and the hyperpolarization phase. Remember that during the absolute refractory period up here, another action potential cannot occur. The sodium ion channels are already closed. But down here, after the cell has repolarized, it can become more positive again if there's a very large stimulus present, which would induce the sodium to come back into the cell. Thus, this is known as a relative refractory period because an action potential could potentially occur. I'm going to post this video on Canvas for you guys. I think it's a really helpful video because it's going to help you visualize the sodium and potassium pump and the action potential. So I will make sure that this is posted in the module for chapter 4. We know that action potentials are constantly occurring in our cells. Action potentials are different from graded potentials in that they cannot diminish over time. So once an action potential is sent down from the axon hillock, it can move down the axon proper to the axon terminal without diminishing. And that's very important because we need that electrical neurotransmission between the presynaptic neuron and the postsynaptic neuron. This is how our neurons communicate. Remember that action potentials are different from graded potentials that are smaller. Graded potentials can diminish over time, and these are sometimes generated by sodium-potassium pumps. However, the frequency of an action potential can change, and that's another reason why they're different. For example, a neuron might fire more and have more action potentials if we're experiencing pain, in comparison to when we feel rested or relaxed. If we look at the top graph here, this is showing us a very low or small stimulus strength. For example, if you're sitting on the couch and you want to simply move your body, this isn't going to require a high frequency of action potentials. Here on the y-axis, we're looking at the membrane potential, and on the x-axis, we're looking at the resting membrane potential, which is sitting at about negative 70 millivolts. As you can see, a small stimulus is going to trigger a lower frequency of action potentials. In graph B, this is showing us a strong stimulus. For example, let's go back to the example with the simple reflex. If you were to step on a sharp object, your body would want to get you away from that pain. When we receive a painful sensation up the spinal cord via the dorsal root, our sensory neurons are going to tell our motor neurons that we need to command the foot to move away from the sharp object. When motor neurons synapse down the ventral root and back down to our foot, they're going to command cells in the muscle at the neuromuscular junction to move and get away from this sharp object. This is going to require a higher frequency of action potentials to transmit this information. This is important because your body realizes that you are in pain, and it's very important to get away from this sharp object. This would be the case of our simple reflex example, whereas image A is just showing us how the body could respond in a less stressful situation. Remember that action potentials cannot move backward, they can only move forward. So for example, if they begin at the axon hillock, right at the bottom of the soma or cell body, action potentials will move down the axon proper, jumping from the nodes of Ranvier, and they will never move backwards. For your first exam, you should know how to label an action potential. So I created this worksheet and I posted it on Canvas for you guys. You can print this out or use it on your iPad or computer and practice labeling all of the steps of the action potential on your own. So first, I'm going to review this image. I want you to watch this video to understand everything and then go and try it on your own at home. So here on the y-axis, this is showing us our membrane potential, and this is measured in millivolts. And then the x-axis is showing us time in milliseconds. Remember that action potentials occur very quickly. So first, we're going to look at A. A is at negative 70 millivolts, and this is known as the resting membrane potential. Remember that the cell is typically around negative 65 to negative 70 millivolts at rest, meaning that it's more positively charged inside of the neuron relative to the outside of the neuron. If there is a small influx of sodium cations that can come into the neuron so that it reaches its threshold, the cell is going to become a little more positive. And at threshold, which is typically around negative 55 millivolts, this is when our voltage-gated sodium ion channels can open. When these sodium ion channels open, there's a large and quick influx of sodium from the extracellular fluid into the cytosol. And this stage here at letter C is known as depolarization, when the cell becomes more positively charged inside. As the cell depolarizes, it's going to reach this optimal or positive charge known as the overshoot at D. So here at letter D, this is about 30 millivolts, and so this is when this voltage-gated sodium ion channels will close. Remember here, this is known as the overshoot, and this is also the time where the absolute refractory period takes place. At this point, another action potential cannot occur under any circumstances because the cell is already too positive. At this stage, once those voltage-gated sodium ion channels close, the voltage-gated potassium ion channels will open. And while they open, potassium moves a little slower, and it's going to slowly leave the cytosol of the cell into the extracellular fluid, causing the cell to become more negative again. This is known as repolarization, and this is done to make the resting membrane potential more negative again. Here, when we have potassium that's exiting, we have this hyperpolarization that can potentially occur at letter F. And when the cell becomes hyperpolarized, it's actually more negative than it is at its resting state. So this letter F here would be symbolizing hyperpolarization. Now here at G, we know that the membrane potential has gone back to its resting membrane potential. This time here is also known as the relative refractory period because the cell has now become more positive. It is closer to its resting membrane potential. It can potentially reach threshold again at negative 55 millivolts. And because of this, a very large stimulus would need to reactivate the cell so that it would reach this negative 55 millivolts to get to threshold. This could be a strong stimulus like stepping on a sharp object that would thus generate this need to fire another action potential. And this goes back to that previous slide I talked about how action potential frequency can change. So a large stimulus here at the relative refractory period could induce another action potential because they may need to be more frequent to respond to the stronger stimulus. So here's our final review of the action potential. You can use this image in addition to the handout on Canvas to understand all of the different steps of the action potential. You should know this in chronological order. You should know at rest the cell sits at negative 75 millivolts, and when sodium cations come inside of the cell, it can reach its threshold at negative 55 millivolts, which is going to make it slightly more positive, which is going to induce the opening of voltage-gated sodium ion channels. The cell will depolarize, meaning it becomes much more positive. And at the overshoot, at about 30 to 40 millivolts, the cell is very positive, which means that this is the absolute refractory period. And another action potential cannot occur right here because the neuron is already too positive. Then the sodium ion channel gates will close, the potassium ion channel gates will open to allow potassium to enter back inside of the cell to try to regain its negative resting membrane potential. Remember that there are leaky potassium gates, so sometimes the cell becomes hyperpolarized, meaning it's more negatively charged than it was during its resting state. And here, this can be known as the relative refractory period, in which the cell can potentially fire another action potential if there's an extremely large stimulus that's going to make it more positive. This is why it's important to understand that the frequency of action potentials can occur more often. So make sure that you understand all of these steps for your first exam, and I highly, highly recommend printing out that worksheet and actually drawing out these steps and practicing, because hopefully this is going to help increase your learning and be able to recall this information for your exam.